Bio 181, Chapter 13 on human inheritance. And we're going to talk about genetic map and a little bit about some heritable disorders and tests. So a genetic map is simply a representation of the position of the genes on a chromosome. So you can see it over here. I just think it's interesting. The human genome has been mapped for a while now. So we have started to figure out at the sequence. So this is a long sequence of DNA, right? That is coiled up. But if you go, along this sequence, you're going to find regions that code for different traits. So you've got the traits for especially genetic disorders. We've spent a lot of time and money investigating where those are so that we can screen for them. So those are fairly well known. And so you can find different sequences on the DNA and how they correspond to different disorders. So that's a genetic map, and it doesn't just have to be disorders, but that's what we know a lot about. Linked genes, this was really useful before in terms of determining the map before we had sequencing. But it's still of interest because if genes are located very close together on the chromosome, then they're often inherited together. So you do have a random separation, and in meiosis and crossing over, homologous chromosomes line up in synapsis and they swap parts. But if the genes are really close to each other on the chromosome, the chances when they get swapped, they'll stay together. So if the genes are close, they're linked. If they're far apart, they're not linked. And so there's a good probability that they won't be inherited together just due to crossing over in meiosis. All right, this topic actually was introduced previously. Non-disjunction. So errors occur during meiosis because of non-disjunction, and this is where they don't, the chromosomes don't separate equally. So usually, they line up during metaphase. So this is showing us the second round of cellular division. If we're looking at meiosis, where the sister chromatids are lined up in the center. And what you'd expect to see is over here on the top, where you have, each sister chromatid is going to be headed to the opposite pole prior to cytokinesis. In non-disjunction, they don't split equally. And so in this case, you can see that the entire chromosome still copied is going to one side, and then you have nothing going to the other side. So aneuploidy is the loss or gain of chromosomes, and this is actually pretty high. So the rate may be as high as 10% of conceptions. Now, that said, they are not going to be viable, and so pretty quickly, through the early divisions, the cell is not going to continue to divide because the loss of all of those genes is just not sustainable in terms of an organism. So what a few examples are shown here in terms of inheritance patterns resulting from non-disjunction. So Turner syndrome, in terms of the sex chromosomes, you have one X chromosome, but you have the absence of a Y chromosome. And these individuals are still able to live and have a fulfilling life. They don't have the chromosomes on the Y, but that's okay because females, I'm just going to erase that. So females only have two X's. And so it really is okay if you're missing some of those genes on the Y chromosome. Similarly, you could have an additional X, and that results in another syndrome. You tend to have elevated secondary female characteristics, but again, still viable offspring, and it's quite common. Down syndrome, we get an extra copy of chromosome 21, trisomy. So when that chromosome split, there's just an extra copy there, and that results in that genetic condition. So this is showing an example of a Punnett square where you might have an unequal distribution of your sex chromosomes. The male is normal up here. This is triple X syndrome over here, which, whoops, it's going to lead to triple X syndrome. But we have a parent, the mother, that has an egg that is already starting with 2 X chromosomes. And so when that is fertilized by the sperm, you end up with triple X syndrome. And again, that's another fairly common genetic condition. So there are a few methods for sampling early on before birth and looking for genetic disorders. Amniocentesis and chorionic villus sampling are 2 examples that you're responsible for. The amniotic fluid is sampled and it contains cells that are from the fetus. This is that are suspended in here, right? So there's been some shedding, and so you can get the DNA of the fetus at that point. This is 15.5 weeks. So a little bit later on, a little bit more detailed than the chorionic villus sampling, which is about 10 weeks or more. And so this is the fetal part of the placenta that the sampling is from. So you can get cells here. And so you can determine early on if there are any conditions that you can potentially mitigate early in the pregnancy. So the last slide is just a few examples of disorders from a table in our book. So sickle cell anemia is something that we'll go over in more detail, as well as hemophilia. So sickle cell anemia is a recessive disorder. It's a very common disorder, and it does have some benefits to it in terms of it helps reduce the prevalence of death from malaria if you're infected with malaria. So the sickling of the cells has actually been conserved in areas with high malaria outbreaks, so some African regions. And then hemophilia, this example is an X-linked recessive trait. So again, this is one that is more common in males. And you can see that over here in terms of the rate. It's really low in females, but because it's X-linked recessive, it's common in males. All right, and that's it for this video.